Yeah, thank you so much. Moving to the next session, and again, we are very excited to have, Nobel Prize laureate Michael Houghton, who will be providing a closing keynote presentation.
Great, and so James here. Can you guys,
yep.
you guys can hear me okay? Yeah.
Yes, James.
So James Taylor here, co-founder, CEO of Precision Nanosystems, and it's a great honor to introduce our next speaker, Dr. Michael Houghton. So Dr. Houghton will inspire us with his extensive vaccine experience, including adjuvanted subunit proteins, DNA vaccines of various formulations and delivery, attenuated viral vectors to deliver vaccines, and the great potential of RNA vaccines in vaccinology. Dr. Houghton needs no introduction, I'm sure, but is an international leader in viral hepatitis and a 2020 Nobel Prize laureate. Dr. Houghton, along with with Q-Lim Chu, George Koo, and Daniel Bradley, co-discovered hepatitis C in 1989. The discovery of hepatitis C led to the rapid development of diagnostic reagents to detect HCV in blood supplies, which has reduced the risk of acquiring HCV through blood transfusion from 1 in 3 to about 1 in 2 million. He also co-discovered the hepatitis B genome in 1986. And it is estimated that antibody testing has prevented at least 40,000 new infections per year in the US alone, and many more worldwide. Houghton is currently Canada Excellence Research Chair in Virology and Li Ka-shing Professor of Virology at the University and Director of the Li Ka-shing Applied Virology Institute. He has received numerous international prizes for his work, including the 2000 Clinical Lasker Awards, the Robert Koch Medal in 1993, and the Canadian CSL Gold Medal in 2012. His passion is the application of biomedical research to prevent disease. And with that, sir, I will turn it over to you.
Thank you very much, James. I hope you can hear me okay.
Yep, sounds great.
Thank you. And hopefully you can see my slides.
Very good.
Excellent. Well, thank you very much to Precision for inviting me to address your audience today. Unfortunately, I have not been able to join your conference earlier, so I hope my comments are not going to be too repetitive from your other participants. But it is my pleasure to talk about what I see as the great potential of RNA vaccines. I'd like to start off just reviewing traditional vaccine technologies. They are very large and have been used for many years in some cases. So I thought it would be useful just to review how vaccines have been made in the past. I think many started off taking, for example, a pathogenic virus and attenuating its pathogenicity by passage in cell culture, passage in animals, to end up with a strain that is safe, doesn't cause the disease of the original virus, but nonetheless invokes a protective immune response. So vaccines included in this category would be the well-known vaccinia virus that protects against smallpox and has eradicated smallpox. Yellow fever viral vaccines, measles, mumps, rubella vaccines. All these have been very successful. A bit more recently from Merck, the chickenpox vaccine based on an attenuated varicella viral vaccine, rotavirus, and polio with the Sabin vaccine. So this category obviously has been very successful. It's very historic and very traditional, and still used and very effective. Then I think the field moved on to whole viral vaccines. Jonas Salk developed the first polio vaccine based on inactivated polio virus, chemically inactivated virus. But we've seen many other examples of that, for example, with influenza and rabies, and also hepatitis A. I think 30 years ago, approximately, recombinant subunit protein vaccines started to emerge. And these typically are used with various adjuvants to enhance the immune response. These also have been very successful vaccines, tried and tested. For example, the vaccine against hepatitis B virus uses a recombinant surface antigen protein derived from recombinant yeast. The human papillomavirus has been licensed for, I believe, around 10 years. And that uses recombinant proteins from this virus combined with powerful adjuvants. More recently, the vaccine against shingles virus, herpes zoster, has been very successful, and this was approved just only a few years ago, and is essentially the envelope protein of this virus made in CHO cells, so completely non-infectious, combined with a very powerful adjuvant called AS01, which is very good at stimulating antibodies, but also cellular T-cell responses required for protection against shingles, especially in old age. It's a very effective vaccine. And in fact, has superseded the live attenuated virus vaccine developed earlier against this virus. Hep A, also there's a recombinant subunit protein with adjuvant formulation developed. And then we have Bordella pertussis, whooping cough, which is part of the DTaP combined vaccine. This involves recombinant subunit proteins, as well as meningococcal B vaccines, which have been developed recently, which all involve recombinant proteins. Historically, some of the earlier vaccines developed targeted the actual toxins from bacteria causing diphtheria, tetanus, and pertussis. And in this technology, the toxins are chemically inactivated. The toxic proteins are chemically inactivated, and then have shown themselves to be very effective vaccines to prevent disease with these bacterial infections. And then finally, the whole field has developed by taking polysaccharides from the bacterial capsule and eliciting antibodies to those molecules. And I would say the breakthrough there has been the concept of conjugating those polysaccharides with T helper proteins, like diphtheria toxoid, tetanus toxoid, etc., to actually help stimulate antibodies to the polysaccharides that provide the protection. Examples of this would be the Haemophilus influenzae type B vaccine, the meningococcal ACYW vaccine, Streptococcus pneumoniae with PCV13 vaccine. And then there's the 23-valent pneumococcal polysaccharide vaccine that is also very effective. So in dealing with vaccines and emerging pathogens, whether they be viruses, bacteria, we have a huge armory to confront these diseases. Now, as I'm sure has been discussed at your meeting, these generally take a long time to actually go through clinical testing and approval. And I think what we've seen with COVID has been remarkable, just the speed at which these vaccines have been produced and tested and are on the point, I would think, of being approved for emergency use. So the new vaccine approaches, I noticed Jeffrey Ulmer is in the audience and a speaker. He was involved at Merck in developing DNA vaccines against various pathogens. If you inject a piece of DNA into the arm of someone, it gets expressed into the protein and you do get an immune response. And Jeffrey was one of the first people to show this technology could really be used for human vaccines. I think that's been taken up by a company called Inovio, who are enhancing the potency of DNA vaccines by electroporating, actually increasing the rate of uptake into myoblasts in the muscle, as well as antigen presenting cells. And although it sounds rather hazardous, I believe Inovio have got the technology down to the point where it's really feasible and user-friendly. And it's particularly good at producing T-cell responses, especially cytotoxic lymphocyte responses. But that is a technology in progress for COVID, and it's going to be interesting to see how that develops. Obviously, the great work from Oxford University, Adrian Hill and Sarah Gilbert and colleagues is looking extremely promising. Also, we're looking forward to some efficacy data coming out of their phase 3 trials soon. And as many of you know, they deliver various genes encoding vaccine antigens via the use of replication defective adenoviruses, and others use replication defective vaccinia viruses. But Oxford have really utilized a chimpanzee adenovirus strain that does not infect humans, and therefore immunity in the population is essentially non-existent. And they can deliver the COVID spike protein through this delivery system. Adrian Hill has been working on this for many years in the context of malaria. And it was really so nice to see them quickly adapt that technology, developed over many years to develop a COVID vaccine. But also, it can be used for other things, for example, influenza, HIV, and many other viral and also bacterial diseases. So these are all, potentially very ubiquitous systems. And I think I've been incredibly impressed with the speed of the RNA vaccine technology. I remember reading papers 20 years ago from Franz Heinz in Vienna, where he presented evidence in animals that if he injected an attenuated RNA molecule from certain flaviviruses, he was able to get a weak replication and a very strong immune response that protected the animals against challenge. And RNA molecules are very well recognized by antigen presenting cells of the immune system. They are taken up very efficiently, which leads to a very good immune response. So 20 years ago, I was expecting this to emerge as a major technology, but I think what happened was RNA is inherently unstable. It's very sensitive to ribonucleases that are circulating in our bodies, and is quickly cleaved and inactivated. So I think the brilliant evolution of this technology has come from incorporating the RNA into a lipid nanoparticle to protect it, while still enabling it to be taken up by antigen presenting cells. So obviously it's being used very successfully so far for COVID. It has been used and published on 2 or 3 years ago on influenza vaccines. As you've been hearing at this conference, there are a few tricks that have to be played with lipid RNA nanoparticles. The first is to modify the nucleosides to inhibit innate immunity. Otherwise, the innate immune response, the body, when it first sees the RNA molecule, responds immunologically and creates a number of cytokines and a number of responses that degrade the RNA, and therefore the RNA is not expressed very well into protein and therefore is not very immunogenic. So it's a credit to the innate immune system that the inventors of this technology have to modify the nucleosides incorporated into the RNA to actually downregulate the innate immune response, thus allowing good expression into protein, thus allowing good immunogenicity. I think the other discovery that inventors of this technology came up with was to cap the RNA. Our cellular RNAs are generally capped at the 5' end, which has been shown to increase translational efficiency. So RNA vaccines, in addition to the modified nucleosides to inhibit the innate response, also incorporate a cap to increase translation efficiency. And then, as I already mentioned, formulating these modified RNA vaccines into a lipid nanoparticle is crucial, first to stabilize the RNA in the body, but also forming a nanoparticle which the immune system recognizes as a potential viral particle. It's around 100 nanometers in size, which is a typical size for a virus. And so the immune system has evolved to detect particles like that in the body and to enhance uptake by antigen-presenting cells, thus developing a good immune response. So I've been amazed at just how rapidly in the last couple of years the field of RNA nanoparticles has developed, and I think we all have, everyone in the community. I just wanted to show you a little bit of data. This is from Dr. Baal, who is at Moderna. And I was interested to see just what clinical data they had on influenza using RNA nanoparticles. So they published this paper where they were looking at an avian influenza, H10N8, potential pandemic strain. It did cross into a few humans with devastating consequences. Fortunately, it never developed into a serious pandemic, but it had the potential to do so. So Moderna, with their RNA nanoparticle technology, did investigate a vaccine to this pathogen. They did animal studies, and I'm showing you here on this slide some data from a human clinical trial, a phase 1 trial that they published in 2017. And in the top panels of this slide, you see the number of recipients that actually responded with an immune response as measured by a hemagglutinin inhibition assay. They gave 2 doses, 100 micrograms of the RNA each time. And you can see that 6 weeks later, on day 43, 100% of patients seroconverted. They had hemagglutinin inhibition titers of at least 40. And on the right upper panel, you can see the data from a microneutralization assay of the virus. And once again, at 6 weeks after the second dose, nearly 90% of people had neutralizing titers greater than 20 in their particular assay. And then the bottom panels show the data in a different way. You're actually looking on the bottom left part of the slide at the hemagglutinin inhibition titers on day 43, 6 weeks after the second dose. The placebos did not have any hemagglutinin inhibition titers, but the vaccinees all responded. There was a range from approximately 50 to 150 with a median titer of around 80. And once again, on the bottom right panel, if the data is expressed in terms of microneutralizing titers, you can see the placebos were 0. And there was a range of neutralizing titers induced in different recipients with a median neutralizing titer of around 50. And these are fairly similar to the kind of responses, immune responses you get using conventional flu vaccines. Now, in terms of safety in humans, they got a pretty well tolerated profile here. The majority of adverse events were mild. 107 out of 163 events were mild or moderate. Moderate adverse events in around 1/3 of the recipients. using the Center for Biologics Evaluation and Research Severity Scale. And they state that the adverse events were comparable in frequency, nature, and severity to unadjuvanted as well as adjuvanted H1N1 influenza vaccines. So this is looking very good. Only 4 events were reported by 3 subjects. So that represents 13% of exposed subjects that were categorized as severe and included injection site erythema, injection site induration, so swelling and some lesions at the site of injection, as well as systemic effects like chills, similar to what we get in the common cold. So the authors from Moderna concluded this reactogenicity profile is similar to that of a monovalent AS03 adjuvanted H1N1 vaccine. And it's comparable to that is observed in meningococcal conjugate vaccines in healthy adults. So I think 3 years ago, we could conclude that this technology was looking very promising. Obviously, they and others, including Precision Nanosystems, are applying this technology to COVID. And the RNA vaccine, in all cases, as far as I'm aware, encodes the spike protein of the virus. This is a very large glycoprotein on the surface of the virion, and it forms a trimer. And on the upper left-hand part of this slide, you're looking at the structure of the trimer, and the green portion on top of the trimer represents the receptor binding domain of that trimer. It's interesting that in the trimer, 2 of the receptor binding domains are actually in the closed conformation. They're actually not exposed, but one of the trimeric molecules has the receptor binding domain actually exposed, and you can see that in the upper left where it's being exposed and is readily accessible to the immune response. Now, Moderna just, I think last week, I believe it was, reported results from their phase 3 trials involving up to 40,000 people. So they have immunized the volunteers 2 times, approximately 1 month apart. And then they followed them up looking for infection events in the controls, placebo controls, as well as the vaccinees. And so they reported interim preliminary data in the follow-up representing 1 to 2 months after the second dose. And to everyone's delight, reported that the vaccine appears to be 94% effective at preventing infection. This has not been published yet in a peer-reviewed journal, but nonetheless, this is extremely good news. Also, they reported that the vaccine is protective not just against infection, but against the incidence of severe disease. And then also in the last couple of weeks, the RNA vaccine originally developed by BioNTech in Germany, who partnered with Pfizer to scale up the production and deliver the vaccine. In their phase 3 trial, they reported greater than 90% efficacy in preventing infection within the first 1 to 2 months following the 2-dose vaccination schedule. BioNTech and Pfizer also reported that the vaccine was well tolerated. There was some injection site pain, some fever, and some headache, but in some ways, when you experience those side effects with a vaccine, it's actually a good sign. It means your immune system is being revved up, it's recognizing the vaccine. I had a shingles vaccine a couple of weeks ago, and I was actually quite pleased that I didn't feel too well for a couple of days. It means I'm responding very well to that vaccine. And then this morning on BBC News, it was reported from BioNTech and Pfizer that not only is their vaccine efficacious in the general population in all age groups, but they've reported, once again, this is not peer-reviewed data, it's just a public announcement, that it's 94% effective in people over 65 years of age. So that is really good news, because certainly I was worried that maybe it wouldn't be as effective in the elderly as they previously reported in all age groups. So this is looking really good. And of course, RNA vaccines can be manufactured very rapidly. Obviously, the great response to the COVID epidemic is an example of that. And also, there is some concern that there might be some mutants of COVID that may be less sensitive to neutralization by the current vaccines. RNA technology gives you the ability to respond very quickly, to modify the vaccine and rapidly turn around an improved version that might be better against any mutants that emerge. And excuse me, as you probably know, in Denmark, COVID transmission has occurred into the mink population. There have been some mutations occurring in the minks, and the virus then has been able to come out of minks and reinfect humans. There is some concern that that virus is less neutralizable to current vaccine formulations, although I have not seen any data categorically showing that. So I think we need to see the data on that. And I personally will be surprised if it's completely resistant to these current vaccines. I think that's unlikely, but we do need to see the data. And in worst case scenario is RNA vaccine technology can respond very quickly to any mutants that emerge. So I see the potential and challenges of RNA vaccines in the following terms. I think, obviously, it is absolutely amazing how fast and how effective so far these vaccines are against the COVID epidemic. And I think the RNA technology has the potential to transform the vaccine industry. I think theoretically it could be used to protect against most viral infections, most bacterial infections, and most parasitic diseases that afflict man. Obviously, it can only be used in the terms of producing proteins encoded by RNA. So we have many very effective conjugated polysaccharide vaccines against bacteria, where RNA vaccines will not be applicable. But most pathogens, RNA vaccine technology can be applied to. So I think it's really going to change the way that vaccines are produced in the future. Now, that said, there are important questions and challenges that remain, and I just came into the end of the discussion by your panel before my talk, and I certainly agree there are some issues that are remaining. The first is the duration of immunity. So far, these great potency data coming from the phase 3 trials is extremely encouraging, but it is only after 1 to 2 months of follow-up, approximately. So a key question, especially with COVID, is, are people protected in the long term? I think from our knowledge of what happens with influenza vaccines and vaccines in general, we can predict the immune response will decline over time. That is normal. That's how the immune system works. And with influenza, for example, and with other vaccines, there's a level of protection that is used to guide the need for boosters. And I think that's probably what's going to happen for COVID. There'll be a minimum titer of antibodies that will be defined as being protective. And when people get below that, they will need boosters. So I think the key question right now is how long is this immunity going to last? And they're following up 40,000 people. So I think they have the ability to answer that. And of course, when the vaccine is rolled out to the general population, the efficacy and the duration of efficacy can be determined over time. But this is an important one. We need to know just how long this lasts. Probably booster shots will be required, perhaps on an annual basis, to keep up antibody titers and T-cell responses. So the question there is, with RNA vaccines, are they going to be well tolerated if you have to immunize people every year? Especially if RNA vaccines are rolled out to other pathogens. So if they're being used against a lot of infectious agents, people are getting maybe, I don't know, 2 or 3 vaccine shots a year. We need to make sure that this technology is safe enough for such a widespread use. The other key question, obviously, is how potent is the RNA vaccine compared with, say, the adenovirus approach of Oxford, being marketed by AstraZeneca. And also how potent are the RNA vaccines relative to the subunit proteins that are being developed against COVID. For example, blanking on the name now, but there are several companies out there developing spike protein from recombinant cells, combining it with adjuvant. This is a tried and tested method and has been shown to give long-lasting immunity. So the potency of RNA versus the other types of vaccines needs to be evaluated, and is. Dr. Shattuck raised a very good point. In terms of rolling out RNA vaccine technology in a general way, cost of goods is going to be very important. Many of the vaccines that we have using traditional approaches are very cheap. And that's very important to healthcare systems, that we don't increase the economic burden. So cost of goods is going to be a big issue in just how much the vaccine technology is rolled out to other pathogens. And then, as many of you know, RNA vaccines do have a little bit of a weakness, I would say, in terms of distributing these vaccines. BioNTech and Pfizer, their vaccine needs to be stored at -70 centigrade. Moderna claimed that their vaccine can be stored at -20, and that's good news. But the adeno-vectored vaccines, they can be stored in a regular refrigerator at +4, as can protein subunit vaccines. So I think for RNA vaccine technology to be extended to other pathogens and to really dominate the vaccine industry, a solution needs to be produced for the stability. We need to have a RNA vaccine that is stable at 4 degrees centigrade for many weeks, if not many months. And I would imagine that's going to be possible, but it needs to happen. I'd like to leave you with one message, one final message, and I'd like to address this question. Would a vaccine stockpiled against the SARS coronavirus 1 that occurred in 2003, if we had stockpiled that vaccine, could it have been used effectively against the COVID-19 epidemic? And I think this is a big lesson that we will learn from the COVID epidemic. The next slide shows some data from Walls, Dr. Walls et al., published in Cell earlier this year. And they were making the trimeric spike protein, as you can see in the top left, this rather huge trimeric complex. And they wanted to, they were actually making it against the COVID-19 virus, as well as the 2003 isolate of coronavirus causing SARS. And what they did is to make the trimeric spike, they immunized mice, and then they asked the question, does it neutralize the coronavirus 1 isolate of 2003? In the bottom left, you can see that the mouse antisera was very effective at neutralizing the infectivity of pseudoparticles derived from coronavirus 1. And if you look over on the right-hand side, you can see we're looking at different mice. And so against these 2003 isolate, all the mice were very effective at neutralizing the infectivity of that virus. There was 100% inhibition of infectivity in cell cultures. And if you look on the bottom right, you see that the mouse antisera, although it wasn't as effective as neutralizing the infectivity of COVID-19, it was pretty good. There was around 90% inhibition of infectivity of the COVID-19 virus. So this is a very important lesson that I think we will learn, and that is, as outbreaks of coronavirus occur, but this applies also to pandemic influenza strains, we must stockpile vaccines, because in the future outbreaks, there could well be some cross-reactivity, enough to provide at least some protection against a new strain. So I think this is definitely a lesson we have to learn from the COVID-19. And that's all my slides. I'd like to thank again Precision for inviting me to address you today, and be happy to try to answer any questions you have.
Excellent. James Taylor here, and I have a question, particularly kind of high level. If you're a vaccine developer and you're agnostic to a platform, you're not coming at it with a specific modality, how do you make a decision on which platform to use against a given pathogen?
Well, I think you have to test the various platforms. And, for hepatitis C virus, which I've been working on for decades, I do believe we have an effective vaccine using recombinant protein subunits and adjuvants. I do believe that we will get at least partial efficacy with the vaccine we're developing. But I think now with the rapid emergence of RNA vaccines, it's possible that technology could either be used in combination with protein subunits, or may even replace the need for protein subunits. And so I think you just have to try the different methods and compare them in animals, and preferably actually compare them in humans in phase 1 trials. So I think that's the way to do it.
Fantastic. And if we solved it with the RNA vaccines, which is obviously a big part of this conference, if we solve the stability challenges, which Peter indicated is probably a point in time, not a long-term issue for these technologies, and then potentially with these being a cell-free based manufacturing, we'll have the ability to drive down costs and the like. Do you see mRNA vaccines then becoming a dominant force in vaccinology in amongst the other technologies? And how do you see it fitting in long term?
Yeah, I mean, I think it's going to be transformative. I think it has the potential to become the dominant technology in human vaccines. I think cost of goods is going to be an issue if we have very good vaccines out there on the market that are very cheap. So then it's going to get down to economic issues. Can you make it cheaply enough? But I think many vaccines are quite expensive, whether they're targeting viruses or bacteria. And I think RNA technology really is going to move into that space and be very competitive. Provided you can show that immunity lasts as well as the traditional vaccines, provided that if it's rolled out into many different vaccines, it can be used safely when people receive multiple doses. And if you can solve the stability issue, which I agree, I think probably you will be able to do that. So I think it's got huge potential. I really am. I think I think potentially it's very transformative to vaccines in general.
Excellent. Well, thank you so much, Michael, for today's presentation and the fantastic work you've done over your long career. And now I'm going to hand it over to Andy Gale.
Yep. And once again, thank you, Michael. Excited to see you coming into the RNA vaccine space in the near future. It'd be great to see what the combination of your work in HCV and RNA can do. So this is just to remind you, the next session, there's going to be a follow-up question and answer session at 12:30 Eastern time, 9:30 Pacific time. Here, list of the panelists, and then we'll move into the closing ceremony and celebration of the 10th anniversary of Precision Nanosystems. So with that, I'll thank everyone, and we'll start up again in about 20 minutes. Back over to you, Jane and Shel. Thank you.
Thank you, everyone.